Hello, esteemed jury member of 3M Talk organized by IAFP 2024. I, Aniket Sharma, second year PhD student at the University of Wyoming Laramie, would like to take this opportunity today to present one of my aims from PhD thesis, which is entitled as to study the impact of biocides on differentially gene expression involved in efflux pumps in E. coli and Salmonella species. Biocides are widely used around the globe for a number of applications. One concern is that such intense usage of biocides could lead to increased bacterial resistance to a product and eventually cross resistance to unrelated antimicrobials. Chemical biocides have been used for centuries in wide applications such as wastewater, foodstuff, and for treating diseases, wounds. But now they have expanded and entered pharmaceuticals, oil and gas, and plastic sectors. Therefore, our aim of the study was to identify bacterial resistance against biocides. Previous studies have shown clinical resistance bacteria isolates have high expression of efflux pump. Therefore, our hypothesis of this aim was disrupted expression of efflux pumps leads to biocide resistance. For this part of a study, the source of samples were bacteria isolated from retail meat and collected from different feedlots previously in the lab. These isolates were subjected to MIC, which is susceptibility testing assay, according to the protocol followed by standardized by CLSI. And we found that more than 50% of E. coli and Salmonella isolates from both the sites were resistant to triclosan, which is one of a biocide. Our understanding of bacterial mechanisms to decrease susceptibility of bacterium against biocides has improved, but they remain poorly studied. There is no doubt that bacteria have a plethora of mechanisms, and often several mechanisms contribute together to the observed resistance phenotype. Our understanding of the effect of biocide interactions with bacteria, and especially on gene expression, remains poor. Therefore, in this part of the study, we conducted in silico experiments where we evaluated triclosan and efflux pumps interactions and found that they have pretty high interactions with the binding energy score of minus 7.6 kcal per mole as depicted in 2D and 3D structures. In future, we plan to validate efflux pump gene expressions via FACS, a phenotypic-based assay, and PCR, a genotypic-based assay. Further identification of mutations will be done via whole genome sequencing, and simultaneously simulation studies will be conducted. Thank you.